So welcome to the evening session and the opening keynote. So it's my pleasure to introduce Marian Walhout as our keynote speaker, our opening keynote speaker this year. And I'll tell just a little bit about Marian. So she got her PhD in biochemistry at Utrecht in the Netherlands. And she did a postdoc with Mark Vidal at Harvard before starting her own lab at UMass Medical School, where she's done amazing work. She's currently the co-director of the systems biology program there. They've really put together an amazing team of systems biologists. Her work focuses on the structure, function, and evolution of biological networks. She has a specific focus on gene regulatory networks. She's pioneered approaches to do gene-centric mapping of regulatory networks. Earlier on, she did important work sort of systematically mapping regulatory networks in worms, some of that in human as well, and really interesting work on understanding the evolution of those by comparing transcription factors in different families and how they differ. More recently, she's done really exciting work that combines sort of at the intersection of regulatory networks and metabolic networks. Cool paper recently on dissecting the metabolic response of worms to differential bacterial diets, which is really exciting. I think one specific point about that work, I think it really foreshadows what's to come in our field in an important way. It really is the intersection of systems biology approaches, system genetics, but taking a mechanistic approach to really dissect that. And I think Marian is really a leader in that, and that work is a nice example of where our field is going. So beyond the great science that Marian has done, she's much more than a great scientist. I think she's an amazing role model. She's been a great mentor. So I've had the pleasure to collaborate with her for several years. She's an amazing mentor to her lab, mentor to young faculty. She was a great mentor to me, even at a different university. And I think really someone that we can look up to. So, and I think beyond that, her community mindedness is worth noting too. She's been a co-organizer of this meeting for several years, several other meetings. She told me once that she doesn't say no when she's asked to do something, which I think is pretty crazy. Maybe that's not true now, but that's actually why she's giving this talk. So let me just end. I like to find embarrassing quotes from people, and I actually found a really great quote from Marian with my Googling skills, which I want to read to you guys. So she said, "Science and art are not too far apart. Dogma and comfort are the enemies, and we have to strive for what I call the scientific Olympics. There's no resting on your laurels. You need to keep pushing for discoveries and insights, as well as training a highly talented group of young scientists from all over the world." And I think that really captures how Marian has approached her science. And I can tell, I think anyone that knows you, so she doesn't do any resting on her laurels, highly passionate about everything she does, and has brought that really to our whole field. So with that, it's a great pleasure to introduce Marian as our opening keynote speaker. Go ahead.
All right, well, it's working great. Thank you, Chad. That was way generous, and I'm glad that you took that as an embarrassing quote. All right, so today is it too loud? What do you guys think? Okay. So I'm going to be talking about gene regulatory networks mostly. My talk is I'm going to try to be high on concepts and low on details and data. And of course, I'm more than happy to talk to anybody in detail about anything we've done in the bar in the next few days. I also want to say, Chad has been saying that she's been doing this and she's been doing that, which of course is so not true. It's an amazing group of people that I have the privilege of working with, two of which are here, Auri, who's in the lab, and Juan, who just started his own lab. So with that, let's get going. All right, so this, for people that work on gene regulatory networks, is really a very well-known picture of early sea urchin development that was dissected in great detail by who you can call the father of the field, Eric Davidson, who unfortunately passed away. And so what you can see here is relationships between transcription factors and genes. Genes are being turned on in a temporal fashion in different parts of the animal and so forth. What you don't see in this picture is that each of these edges in this network is the career and life, at least for 4, 5, 6, 10 years, of one student or postdoc. And so it became clear very early on that this was not sustainable to map out large gene regulatory networks. And with the advent of whole genome sequences and technology development, we and others have been trying to get at a little bit higher pace. And we can argue as we get toward the end of my presentation whether we're succeeding, yes or no. Oops. All right, so there are many systems that have been used for gene regulatory network characterization. Our KR are not on it. I'm sorry, Amy, you gave an amazing talk. Yeah. E. coli, yeast, Arabidopsis, worms, flies, and humans. And today I'll just talk about 2 species, which is most of my talk is going to be about the nematode C. elegans, which has 20,000 protein-coding genes, as do we humans. And then whereas humans, this is, I think, Sarah Teichmann's lab, who predicted that there are about 1,500 transcription factors encoded by the human genome. John Rees-Hoyce in the lab predicted that there are about 1,000 encoded by the C. elegans genome. And so you would think that, conceptually at least, that this is a straightforward problem. You have 20,000 genes, 1,000 or 1,500 transcription factors, a bunch of cells, and you have to fill out this cube, if you will. But in the last 15 years or so, as the field got started, it became more and more clear that it's actually this kind of a picture is what we have to look at, where you don't really know what's up or down. This is one of my favorites from Escher, who was Dutch. And so the reason for this is that these networks are very, very complex. Firstly, there are many types of regulatory regions, promoters, insulators, repressors, enhancers, etc. A big question is how to assign these regions to a target gene, especially in larger and more complex genomes such as that of ours. Of course, there's many different cells, many different conditions through which an animal develops homeostasis, physiology, aging, and disease. And what has become very fashionable more recently is to incorporate genetic variation into gene regulatory networks. And so we had this textbook view on transcription. As Chad said, I was trained in biochemistry and actually worked on transcription in vitro, very painful cold room stuff, assays. And this was really the idea. It's still, we can talk about this toward the end, it's still the idea. Transcription factor binds to DNA, something happens to the target gene. And so about 15 years or so ago, maybe 10 years, the idea was really, if we know all physical interactions, we will know and understand gene regulatory networks. Because physical interactions were equated with regulatory interactions. And that's actually the main point I'm trying to make today. If there's one point you want to take from this, it's this, is that we may have to rethink that. And so, as I said, over the years, many high-throughput techniques have been developed and used. And so the first thought was, if we know where each transcription factor binds in the genome, then we'll know its targets. And so techniques that have been developed and used, as we heard from Anne earlier on, by far the most has been chromatin IP. Then Bas van Steensel in his lab has developed and applied DAM-ID, and we, as I'll talk about, have used sort of a complementary approach by using yeast-1 hybrid assays in yeast. Other people also have the same thinking. If we know the binding site of each transcription factor, we'll search the genome and know its targets. That was going to be so easy. Protein binding microarrays, which were pioneered by Martha Bullock and heavily used also by Tim Hughes, high-throughput CLEX by UC Taipale, bacterial 1 hybrid assays to a lesser degree by Scott Wolf and others. And then it turned out that binding sites are everywhere. So then the thinking was, if we know the binding site of each TF and we know which genomic regions are accessible, where the transcription factor can land, we will know its targets. And so this has been pioneered by defining those open regions by DNase 1, John Stamatou, Yiannopoulos, ATAC-seq by many, many people, including Howard Chang and Bill Greenleaf. All right. And so this is a slide that I stole from, I didn't steal it, he just gave it to me from Juan. Which is really what the thinking is here. It turns out that these approaches are very complementary. And so this is a cartoon of the Indian elephant, as you probably all know. And depending on where you look, you might think the elephant is a rope, it's a wall, it's a spear, it's a fan, etc. And so another take-home point is that really Juan has very nicely shown that these methods are highly complementary, and we should probably employ all of them if we want to get a deeper understanding of physical interactions between transcription factors and the genome, as said here. All right. Now, when you think about it, there are two conceptual ways with which you can approach this problem. You can take transcription factor-centered or protein-to-DNA approaches, or you can take gene-centered or DNA-to-protein approaches. And the vast majority of the field, most notably ENCODE and ModENCODE projects, have used these TF-centered or protein-to-DNA type approaches. And I'll illustrate that here. So if you think of a gene regulatory network as a giant matrix where you have all the regulatory regions and all transcription factors, then what TF-centered methods do is they probe a row in this matrix by asking one transcription factor at a time, what are the genes or loci or enhancers or whatever that this transcription factor associates with. Of course, here you need good antibodies for transcription factors. You would have to redo it in diseases with different mutations, maybe in transcription factors, and repeat it for every single tissue or cell type you want to look at. And indeed, only about 10% or so, even after a long, whoa. Did it die? No, I'm good. Thank you. And so I was going to say something about those consortia and the whole thing went. In any case, so even after 15 years of spending a lot of your money, we have about 10% of transcription factors that have been probed by these approaches. And so we in the lab have pioneered conceptual and practical approaches to go sort of the other way around by taking gene-centered approaches in which we probe a column in this matrix. By asking for one regulatory region at the time, you can see here the gene is the same. What is the repertoire of transcription factors that can bind to that region? The method that we use is called yeast-1 hybrid assays. I'm not going to go into a lot of technical details. Suffice it to say that you have a single hybrid protein, a transcription factor fused to the heterologous GAL4 activation domain. Each regulatory region is probed twice within the same nucleus of yeast. And the readout is both growth and a colorimetric assay. And Bart de Planke, who was my first postdoc, made this system high throughput by combining it with gateway cloning. By no means did we invent the yeast-1 hybrid system, by the way. It's Ira Herskovich, Stan Fields, and others. And then Vanessa Vermeijersen and other people made this system already a little bit higher throughput. And then again, John Rees-Hoyce, when he was in the lab, developed a high throughput platform to start scoring those interactions between regulatory regions and transcription factors. So briefly, the way this works is we plate each transcription factor in quadruplicate, as you can see here, and then we plate that on a plate, yeast-1 mating type. Then we have our DNA bait, we mate the two, and then through a step of replica plating, similar as to SGA analyses, we go to a readout plate that is then automatically analyzed by software that was actually developed mostly in collaboration with Chad. Everything is collaboration with Chad, pretty much, which is why I'm here, really. Okay. And so the first vignette that I want to tell you about is a paper that we published about a year and a half ago, or maybe 2 years by now, that was really led by Juan, who is here, and I encourage you to meet with him if you have a chance, in which we took our human platform, and I should also say that Siobhan Brady made a Arabidopsis platform, and Bart de Planke made a Drosophila and a mouse platform as well. And he tested human enhancers and mutations, and I'll tell you a little bit about that. So firstly, Juan took 246 human developmental enhancers that were defined by Len Pinocchio's lab by sticking them in front of a reporter gene in mice. And then by screening in these yeast-1 hybrid assays, he identified more than 2,000 protein DNA interactions involving 283 TFs. And so here's another take-home point for you. So by doing it's a huge amount of work, but by a relatively small scale experiment, we already touched on way more than 10% of transcription factors, illustrating the complementarity of the approaches. Much, much smaller on the DNA side, of course. One of the things that Juan discovered here is that you can find transcription factors that are similar often, as Chad was saying, that are bound by overlapping set of enhancers. And what Juan could show was that rather than the knee-jerk reaction that probably half of the room has right now, which is that it's all false positives, Juan could really show examples of where these transcription factors can work redundantly or where they can even have opposing functions depending on where you are in development. Say, this factor comes in, represses, then this factor gets expressed, starts to bind and activate so that you can get certain types of regulatory patterns. Another point is what Juan showed is that the transcription factor expression pattern and level are not as big of a deal in one hybrid assays because you just express every single transcription factor under the control of the same promoter in yeast and the same incubator, etc. Whereas CHIP is much better suited for highly expressed transcription factors or broadly expressed transcription factors. One hybrid, of course, also has disadvantages. It doesn't work for heterodimers as of yet, for instance. And then Juan went a step further in what I think is a beautiful set of experiments by asking if we could use this platform to test disease-causing mutations. And he tested both coding mutations in transcription factors as well as non-coding mutations in regulatory sequences that have been shown previously to be causative of different types of human diseases. And so here I'll show you one example of a coding mutation in this transcription factor LHX4. The mutations are indicated here. And now what you can do is you can say, I'm just making quadrants for, say, wild type, mutant, mutant, and so forth, and test them all side by side, same experiment, same incubator, same person, same everything. And what Juan found was that you can see different types of agetic interactions, as Mark Vidal would call them, and I hope to hear more about that on Saturday. And so what you can see here is that for some mutations, you mostly retain interactions, whereas for other ones, you lose interactions completely, and sometimes you also gain interactions. And this can really start to help explain why and how those different disease-causing mutations do cause different diseases. The same is true for those non-coding mutations. Juan found differential interactions for 109. So now each of the plates is different. You have a wild-type plate, and then you have a mutant plate. You do them side by side against all wild-type transcription factors. And you can compare it directly, really apples to apples. And here we found many interactions are lost, and also a third or something that are gained. And Juan went deeper to show the functionality of some of these and the correlation with the gain or loss of transcription factor binding sites. Whilst I sip water, I'll let you look at this. This is the most complicated part of the whole talk. Okay. And so then Juan asked whether this is, what this means, is this meaningful at a sort of meta level? And so what he devised was this scheme of looking at concordance or discordance between a gain or a loss of an interaction and changes in expression of the target that was known actually for these. And so you can be concordant if the expression of the target goes up and you gain an interaction with an activator or you lose an interaction with a repressor. Similarly, if the expression is lower, you can have a gain of an interaction with a repressor or a loss of an interaction with an activator. And as you can see here, this is a little painful to look at. You can just see that the numbers in yellow are much higher than the numbers in green, and the yellow are the concordant ones, and then these are the p-values here, indicating that at a sort of meta level, this starts to make a lot of sense. And so moving forward, now that Juan has his own lab, he's going to use this system to dissect the biophysical impact of genomic variation on transcription factor binding, which I think is very exciting. All right, enough about people. So the rest of the talk, I'm going to talk about C. elegans. So for those of you who are not familiar with the worm, C. elegans is a hermaphrodite. They have each about 250 babies, believe it or not. A very compact genome, fantastic resources and techniques, some resources that are really essential that were developed in Mark Vidal's lab, such as an Orpheome, RNAi resources, etc. The animals are transparent, so what you can do is you can stick a reporter gene in the animal and watch transcription effects in a living animal as it just goes about its business. They develop in about 3 days, live for about 2 weeks, and it's very cost-effective, etc. And the power of this, I hope, will become more clear as we go to our in vivo studies. So few vignettes of what we've learned from C. elegans in the last 14 years or so. So first, Bart de Planck, again, who I mentioned, mapped the first gene-centered network for tissue, in this case the intestine, tiny network at the time, but still. Natalia Martinez, when she was a graduate student in the lab, discovered feedback loops between transcription factors and microRNAs in a fantastic collaboration with Victor Ambrose. Efsun Arda was really the pioneer of a lot of work that's going on in my lab right now when she was a graduate student, and she discovered that nuclear hormone receptors in C. elegans, that's estrogen receptor, vitamin D receptor, and so forth. You and I have 48. C. elegans has about 275 of these, and we'll get back to that later. That these nuclear hormone receptors bind often metabolic genes. And then finally, and Chad alluded to this, John Rees-Hoyes discovered that highly similar transcription factors that are very similar in sequence can still differ very unexpectedly in what we call their interaction profiles, whether it's protein-DNA interactions or protein-protein interactions. And so last year, Juan completed a study that was really a collaboration between a large number of people in the lab, in which we provided the largest gene-centered protein-DNA interaction network that we had mapped to date. And so this network contains about 20,000 protein-DNA interactions between about 2,500 gene promoters and about 366 transcription factors. Note that this is about, close to 40% of all C. elegans transcription factors were touched in this network. He then looked at the quality, the technical quality of this data, and found a very nice overlap with known transcription factor binding sites that were mapped by Tim Hughes' lab, as well as with chip interactions that were detected in the ModEncode project, although this overlap is a little bit more modest. And so then I want to give you just one example of what you can do with this type of data. I don't want to torture you by going through all the figures. But I think what is a nice aspect here is that Juan was able to start predicting which transcription factors are activators and which transcription factors are repressors. So far in the C. elegans literature, it's one gene with one transcription factor, and now this is an activator. And so the reasoning that he used was that if you look at a set of expression profiling experiments for C. elegans and you take that all together, you can start to see if a transcription factor expression level correlates with the yeast-1 hybrid target expression levels. And you might postulate that that transcription factor is therefore an activator. Conversely, if you see sort of an anti-correlation, you might postulate that this is a transcriptional repressor. And so by randomizing the yeast-1 hybrid data, we showed that these associations that we found, 28 predicted activators, 16 repressors, is statistically significant. And here they are. There's a whole bunch of activators. Here are some examples of those factors that there's one interaction in the literature and now it's a repressor or an activator as indicated here. Juan then went on to do quantitative RT-PCR experiments in transcription factor mutants and found for some there's a nice correlation. This is the targets, this is the non-targets co-expression value. And here you can see that the targets of CH38, that's a homodomain transcription factor, go down when you lose the transcription factor. Here, one goes up, two go down, so it becomes a little bit more difficult to interpret. For instance, it could be that this transcription factor can function as both. And so then we went on to integrate Juan's data with John's data from his protein-protein interaction network study between cofactors and transcription factors. And we found that known in the literature, activators tend to bind co-activators, whereas known repressors tend to bind co-repressors. And we asked if the same was true for the predictions that we made using our yeast-1 hybrid data. So here's the network of those factors that we predicted to be activators or repressors and for which we had protein-protein interaction data. And as you can see here, again, indeed, we see a nice correlation between what we predict is a co-activator and physical interactions between, is an activator, and physical interactions between co-activators. And for repressors, the same, a little bit less pronounced. All right, so to go back to this, over the last 15 years or so, many people, some of whom I already mentioned, mapped lots of physical interactions. But it's really been sort of the gorilla in the room, is that how you say it? By not really asking the question of how predictive these physical interactions are of function or regulation. And so we wanted to start to really ask this question and get to some insights. And so again, we come back to our framework of two complementary approaches that you can take conceptually. On the one hand, you can take transcription factor-centered methods, protein to DNA. For instance, you can mutate or do RNAi or drug inhibit a transcription factor, and then look by quantitative RT-PCR or RNA-seq or microarrays at target gene expression. And the study was mentioned before by Fritz, but Frank Holstegen published a very nice study where they did microarray expression profiling of many, many yeast mutants, including all predicted transcription factors. However, you cannot relate those interactions, those regulatory interactions that you find, to physical interactions, because you don't know where they occur and how to really put those two together. And so we asked, can we come up with a strategy to take a promoter, identify the regulatory interactions, and then take the same promoter, identify physical interactions, and see how well they correlate? And so this was an idea that Leslie McNeil, a fantastic former postdoc in the lab, had. She said, okay, what if I use a small set of co-expressed gene promoters and I map regulatory interactions and physical interactions? Maybe I can get a first-pass picture of information flow in a single tissue. That was really the larger objective. And I can also start to address this question of functionality in physical transcription factor DNA interaction networks. So here she is. And so again, we focused on the C. elegans intestine. This was just very practical here. The reason here is that, so firstly, it functions both as gut and liver. We'll come back to that later. But importantly, it's a large tissue, so it's visible by light microscopy. And so if you have a green fluorescent protein in the intestine, you can just look at it after you perturb a TF to see if GFP goes up or down. And as we also tested experimentally, it works very well by RNAi. So we had a bunch of different GFP strains, and you do GFP RNAi, and when you do that in the intestine, GFP goes away. And so Leslie took a set of 19 C. elegans transgenic animals that expressed GFP in the intestine and elsewhere, and performed systematic transcription factor RNAi, and then visually examined changes in GFP only in the intestine. That's an important point. And so details here, if you care. And so she tested most transcription factors, but this is an important point because this will functionally link the effect of a transcription factor to a 1.5 or 2 kb piece of DNA. So the resulting network, so this now really is a gene regulatory network, looks like this. Here we have, and of course you don't have to look at it, the promoters and then transcription factors that regulate more than one promoter and those that regulate only one in this setting. And this was really mind-blowing to me because she found 411 interactions with 177 transcription factors. Now, she did this only at the L4 young adult stage of C. elegans. These animals are not challenged. They're not perturbed. They're not developing. They don't have any environmental insult, nothing. And even then, 177 transcription factors contribute to the activity of these 19 promoters. And even when you look at these are the numbers per promoter, and the median is 18. So for a given promoter, chances are that you find 18 different transcription factors contributing to its activity when you're not doing anything to the system and no dynamics. All right, so here we go. Then Leslie mapped physical interactions using the yeast 1 hybrid platform that I just explained to you, as well as mined physical interactions from the ModEncode project. And what she saw was a relatively modest overlap. And so if we focus just on those transcription factors that were recovered in both assays, you can see that the overlap between the two is indicated in green, physical only in blue, and regulatory only in orange. And you can see that while this is statistically significant, whatever that means, for 1 hybrid assays, it doesn't even get there for chip assays. And actually, as I was showing this slide to my husband yesterday, I had like an epiphany, because you could say, if you want to be nasty, which I don't, but I'll just say it, is you could interpret this as chip having way more false positives, because we know that you find a lot of hovering interactions by chip. But of course, yeast is bad. All right. So, what do we learn from this? Many physical interactions have no regulatory effect in the intestine. That's a very important point. They could still do something somewhere else, right? And for sure, many regulatory interactions are indirect. And so, to capture these indirect interactions, we used actually Carles Pons, who is here, who has done a lot of work with us in collaboration with Chad, used a method developed by Julian Markowitz that is called nested effects modeling. And it's basically epistasis in silico, where you look at the target genes, the RNAi target genes that are shared by transcription factors. So if transcription factor A has 5 targets, 3 of which are shared with B, then B is placed below A, and so forth. And so this is what that hierarchical network looks like. And so this is a network that describes the information flow between transcriptional regulators in the intestine at the L4 young adult stage in C. elegans. I won't go through all the biology here, but there is a lot. L2 is the master regulator of intestinal development and also of maintenance, as Jim McGee has proposed and shown before. And sure enough, it's important for every single promoter. And you can go all the way down through these different paths to these transcription factors down at the very bottom. And so in the beginning, people asked us, and we were also nervous, Leslie and I, on whether we were going to get anything useful with only 19 promoters. But it's incredibly labor intensive, right? 19 promoters doing, I think it's like 50,000 experiments looking by eye. And Leslie, I'm always proud of this, has callus on her nose here from staring through the microscope. That's good. All right. And so what we asked, though, is how robust this network really is. Did we do a good job by taking 19 promoters? And so this, again, was done by Carles. What he did was he took out one transgene, or one promoter, I should say, and all the interacting transcription factors, and then redid this nested effects modeling and counted how many edges of the original model were maintained. And as you can see here, for each of the promoters that you remove, you keep more than 50% of the edges in that network. So it looks really good, and we have some other stuff that I'm not showing. All right, so to come back to this, the question now, of course, becomes, when we really had to face it and ask, are the textbooks wrong? Have we been looking at this picture in the wrong way? And I really hope not, but you could make a very strong prediction that if there's some truth, or a lot of truth, of course, to this, I'm just being facetious, this is true, how can we explain our data? And so the expectation that we thought of was that at some point, you have indirect. At some point, something has to happen at the promoter physically. That has to happen. And so what we asked was, at the end of a path in this network, do we detect now an enrichment for physical interactions between transcription factors and those promoters? And so the way this works is this. For instance, here you have a hierarchical micro-nano network, transcription factor 1 to 4, nothing below 4 to go to gene C. So this is a terminal node for gene C. And we ask, is this more often a physical interaction, yes or no? And it turns out that this is the same plot, but now for the real data, that yes, indeed, we do find an enrichment there, because half of the physical interactions in this network are at terminal nodes. And so I think we are learning, I would argue that we have to start really thinking about these problems a little bit differently and a little bit more with an open mind. Because we learn that binding doesn't have to mean regulation, and we learn that regulation doesn't have to mean binding. And so in the beginning, we thought transcription factor regulates, transcription factor regulates, transcription factor, classical cascades. But that doesn't seem to be the case, because we don't see an enrichment of physical interactions within the hierarchical network model. And so this has to mean that there are other types of regulators that play a role here, such as RNA binding protein and kinases, which I won't talk about. And so moving forward, the questions really are, why do transcription factors bind DNA without affecting transcription? What is needed for a transcription factor to exert a regulatory effect? How do other types of regulators fit into gene regulatory networks or into this hierarchy? And how does cell non-autonomous regulation work, which we had a lot of in this network, but I'm not talking about that now. I am talking about this. And so I'm going to hope to convince you, as I don't have to do so much anymore after Amy's amazing talk, that at least in the C. elegans intestine, there's a large interplay between metabolism and gene regulation. And so we know that those two are intimately related from some sort of small-scale examples that Mike Springer alluded to as well. The lac operon, of course, famous in bacteria, the activation of the Loire pathway to use galactose as a carbon source in yeast, biosynthesis of cholesterol in mammals. And so we wanted to know, what about C. elegans? Does metabolism play a role in gene regulation at a quote-unquote systems level? And so as I said before, the intestine functions as a metabolic tissue. It's both gut and liver in the animal. And we had discovered that there are massive transcriptional changes in response to diet. So that's a metabolic input, if you will. And so this is work done again by Leslie McNeil, and I'll take you quickly through some of the key observations here. So Leslie discovered that if you give different bacterial diets to C. elegans as food, that you can start to see dramatic differences in life history traits. For instance, when you give, this is the sort of standard diet for C. elegans that has been started whenever Sidney Brenner introduced the model as a major organism for biomedical research. Is a strain of E. coli called OP50. And what Leslie discovered was that when you use this soil bacteria, Comomonas DA1877, I'll just call it Comomonas, now the animals develop fast, have fewer babies, and live much shorter. By then doing expression profiling in the worm, when you give them these two different diets, Leslie found changes in many genes. This is just the downregulated when you give them Comomonas. And interestingly, a large chunk of these are metabolic enzymes. So this indicates that you can get metabolic network rewiring when you give the animals a different food source. And then here is this one gene that I'll talk about first as a proxy for the measurement of this transcriptional response, and I'll get to what it does a little bit later on. It's called ACDH1. It's an acyl-CoA dehydrogenase. And it goes down about, depending on the method, a few hundredfold on the Comomonas diet. And it was also one of the promoters that Leslie used in the study that I just told you about. And we know it's regulated by about 50 different transcription factors in the intestine. And so what we found was that also this changes in mRNA levels is caused by transcriptional mechanisms because it just requires 1.5 kb of promoter DNA, this transgenic strain. You can see GFP is on E. coli, and it goes down a lot when you give the animals Comomonas. So something in the diet does something to the worm that does something to transcription to make this difference. And so the question is, how does the Comomonas diet repress this promoter? And before I go into that, I should say that when you mix these two diets, you need only a tiny, tiny, tiny little bit of Comomonas, 1 in 100,000, to make GFP go down dramatically, indicating that Comomonas is quote-unquote dominant, and it makes probably some, as I'll show you, some micronutrient that exerts this effect. And so the three questions that we asked are, how do worms respond to bacterial diets? And to answer that question, we did extensive forward and reverse genetics in C. elegans. How do bacteria elicit these responses? And for that, we did bacterial genetics in both species that we used. And then finally, what is the function of this metabolic network rewiring that occurs at the level of transcription? And for this, we did a synthetic genetic screen and some metabolomics light. Okay. So first, Emma Watson, who was a phenomenal graduate student in the lab, in collaboration with Leslie, did the C. elegans genetics. And what they found was, or what they did, is they looked for genes that when you mutate them in C. elegans, when you do RNAi, now the animals can no longer respond to the Comomonas diet, GFP stays on. And much to our surprise initially, we found that these encode metabolic enzymes, not transcription factors. And I hope that this will become clear as we go along. And so that means that what we found now is that transcription factor affects metabolism, because we know that we get these metabolic genes that change in expression upon this diet. But also that when you mess with the C. elegans metabolic network, this transcriptional effect can no longer occur. So these networks really talk to each other extensively. And so the four different metabolic pathways that are connected in a network that Emma and Leslie discovered are branch-chain amino acid breakdown, leucine, isoleucine, and valine, the TCA cycle that we heard a little bit about, the glycine cleavage system, and the methionine SAM cycle. And so next, Emma first solved the puzzle of what it is that Comomonas makes that elicits these life history trait effects as well as the transcriptional effects, as indicated here. So Emma and Leslie were an amazing team working together. And so Emma decided to screen the E. coli deletion collection and look for GFP effects in the worm. And that's actually how we solved it. And if we have time, I'll be happy to explain you how that worked, because we were looking for a Comomonas compound. And Emma discovered it by screening E. coli. And so in E. coli, you can look for mutants or the precise gene deletions that in C. elegans make GFP go down or up. And then Leslie, at the same time, did a forward genetic screen by making a collection of Comomonas mutants using transposon-based mutagenesis. She had about 5,700 different mutants and looked for those in which GFP can no longer be turned off in the worm. And what we found, I'm not going to again bore you with all the details, but what we found was that the compound that is provided specifically by Comomonas and not by E. coli is vitamin B12, which nicely rests in this network that I showed you earlier. Now, vitamin B12 is an essential cofactor for us. Or vitamin, of course, as it says. It participates as a cofactor in two metabolic reactions. Firstly, the conversion of homocysteine to methionine in the methionine-SAM cycle. And secondly, in the breakdown of propionate or propionyl-CoA, I'll just call it propionate from here on, in which methylmalonyl-CoA mutase uses vitamin B12 as a cofactor. And importantly, mutations in MMCM1 were also found in the C. elegans genetic screens. Now, this is an important point that I have to make here. It turns out, as we went along, that we were looking at the problem upside down. It's not that Comomonas repress, or B12 represses these ACDH1 promoter and others. Rather, it's that E. coli activates it. But E. coli has been the denominator for 40 years, so that's how we looked at it for a long time until we figured it out. And so what happens is that if you don't have B12, or if you can't use B12, propionate builds up and now activates ACDH1 and other genes. And so altogether, what we've discovered here is that bacterial metabolic networks, in this case Comomonas, can provide B12 that talks to the C. elegans gene regulatory network, and this communication is affected when you either perturb the C. elegans metabolic network or you supplement propionate, and then these transcriptional effects are counteracted or reversed. And so the next question that Emma solved is, what is the function of this metabolic network rewiring? So B12 serves to break down propionate. Propionate, when it builds up, activates ACDH1. And so ACDH1 is an acyl-CoA dehydrogenase, as I said. These are known to function in the first step of fatty acid beta oxidation or in branched-chain amino acid breakdown. And I'll summarize all this work that Emma did in just one slide. Here she is again. What Emma discovered is that C. elegans has two breakdown pathways for propionate. It's the first example, as far as we are aware, of an animal having two breakdown pathways for this metabolite that is toxic when you build it up, as exemplified by people that suffer from propionic acidemia. And so on diets that are high in B12, C. elegans uses the canonical breakdown pathway that we also use. But when B12 is low, or when you have mutations in this pathway, propionate builds up, and there's transcriptional activation of 5 genes that altogether comprise this propionate shunt or alternate propionate breakdown pathway. And so this is the function of this transcriptional rewiring of metabolism, and as far as we are aware, is the first example of metabolic network rewiring to prevent the toxic buildup of an endogenous metabolite. All right, so now, to come back to the original question of gene regulatory networks, now what I've shown you is that we know that metabolic genes, when you mess with them, affect transcription in the C. elegans intestine. And the question is, how? And how extensive is this, etc. And so this is the framework that we are working with now in the lab. For instance, I'll stick to the example of ACDH1 and propionate and MMCM1 because it nicely illustrates the way I hope you agree, the way we are thinking about this right now. So MMCM1, which is expressed under the control of some transcription factor that we don't know, the function of this enzyme is to limit levels of propionate. However, when these levels build up, when this is mutated or you don't have B12, propionate now activates or talks to, or whatever the word is, to transcription factors that activate ACDH1. And importantly, when we think back about our hierarchical network model, it's this edge that we could see. And so then, now you would understand that, that there's all these other layers in between. And moving forward, this is what we hope to figure out. And so now, Sushila, I can't possibly, sorry, Sushila, I pronounced your last name. Sushila is testing all 19 reporters and doing systematic RNAi against metabolic genes. But before she could do that, we had to know what the metabolic genes are in C. elegans, which really wasn't understood very well. And so Shafak Yilmaz, a bioengineer in the lab, decided to reconstruct the C. elegans metabolic network. And we can use this network now. I'm not going to tell you much about it, to convert in silico bacteria into worms, which is pretty cool. But here's the details on the network that we have. We have 1,273 genes. We call it iCell 1273. About 600 or so enzymes, 2,000 reactions, and about 1,000 different metabolites in this network. And so now we have a set of predicted metabolic genes. We made RNAi resource of this, and Sushila has started screening and is very actively screening still. I'll just give you one example. Again, this is ACDH1 GFP. You can see here is just vector alone. Here's the GFP RNAi. The fluorescent goes away. And then we have 127 metabolic genes that turn on GFP, and 91 that turn off GFP. And so we're thinking moving forward about 3 possible models by which metabolism can affect transcription. One is a metabolite can directly or indirectly talk to a transcription factor that regulates a gene. Another model is that a metabolite can talk to chromatin, for instance, acetyl, histones are acetylated, methylated, and so forth, which could change in accessibility, for instance. And then there's some examples in the literature in which a metabolic enzyme, ARG56, for instance, was discovered by Mike Snyder, can itself bind to DNA and regulate transcription. What we are doing now to fill in those edges systematically between metabolism and gene regulation, we are performing transcription factor RNAi under a condition where a gene is up or where a gene is down. And so in this case, for instance, what Jyote Bulcha in the lab, together with Gabby Gies, has done is to take either a metabolic gene mutant crossed into the transgenic strain. So I see how awesome this is, that you can do this stuff in C. elegans. Or what he did actually was take the strain, throw a bunch of propionate on it, then do TfRNAi and see which transcription factors you need for this response to this metabolite. And the only thing I can tell you now, it's very much actively going on, is that so far he has discovered that 2 of the 50 transcription factors that we had found before are indeed responsible for mediating the transcriptional response to propionate. How this works, we as of yet have no idea. And so moving forward, we really want to keep asking this question: how do metabolic and gene regulatory networks interact at a systems level? Where and what in metabolism affects where and what in gene regulation? Of course, we're still working with 19 transgenes, and that's just a practical limitation. We can't really go high throughput, although I would love to. Longer term, and if somebody's interested in joining the lab to start working on this problem, we also hope to fit in RNA-binding proteins, kinases, phosphatases, chromatin factors, and so forth, to really put all of this information together into what we call multi-parameter networks, in which we combine different types of networks, different types of nodes, all together illustrating the information flow, if you will, regulatory information flow of gene regulation in the intestine. And with that, as I said in the beginning, I am very fortunate to have worked with and still work with an amazing team of people. Bart de Planke was the very first postdoc who started the One Hybrid work. He's now a tenured faculty at the EPFL in Lausanne. John Rees-Hoyes moved on and is at Novartis right now. Efsoon was a student, is now at the NIH. Natalia was a student, now at the NIH. Leslie McNeil, phenomenal postdoctoral fellow, now at the faculty at McMaster in Canada. Juan, who's in the room, and I encourage you to talk to him about the human data that we have in the project, who's now faculty at BU, and Emma Watson, who is now a postdoctoral fellow in Steve Ellich's lab. And this is the team that we have now. Sushila showed you a little bit of what she's working on right now, as well as Jyote and Gabby. Shafak is absolutely indispensable for everything we're doing now, which was just a stroke of luck. And then finally, Aoi, who's in the room as well. Since I was going to do this on Tuesday, I was going to encourage you to look at her poster. But now I'm just going to encourage you to go find her and talk to her about her exciting work, in which she has discovered bacterial metabolic pathways that affect how C. elegans can respond to different chemotherapeutic drugs. And we hope to expand on those studies moving forward as well, because we find those interactions fascinating, but also we think that they might help to start making a framework for how the microbiota can affect positively or negatively a cancer patient's response to chemotherapeutic drugs. And of course, Chad. Should I do now like a whole Chad thing? No. Amazing. Collaborations are fantastic. We love working with him. Carles, that was such a great collaboration, many collaborations with Juan and with John and with Leslie, etc. Juan and I also collaborated extensively with Needy, who's also in the audience, who was a postdoc in Mark Vidal's lab now, as you heard earlier, is a faculty in Texas, and Dave Hill, who's just overall awesomeness, is on this slide as well. And then finally, before I take any questions, and I hope that you have some, I think we should at least say happy birthday to Pascal. I know. Happy birthday. I'm not singing.
Thank you, Marian. So we have time for some questions. Maybe I'll get started with a question. Oh.
Thank you. Very nice talk. So
Use the mic.
yeah, very nice talk. Thank you. I'm wondering about this overlap between physical and regulatory interactions. So how many different transcription factors you studied? And I imagine it was many of them. And did you find subgroups of transcription factors that had higher or large overlaps,
Yeah, that's a great
versus the small overlaps?
Yeah, that's a great question. So I think your question is very important. I think our data set is too small to give a real solid number and to try parsing out different types of transcription factors. When we did the statistical overlap, we only used factors that worked in both assays, so we could really compare apples to apples. We can also talk about why, when a factor binds, doesn't it have an effect? One potential mechanism, and Juan has had some data for that, is redundancy, of course. But then we do find 18 interactions in the median per promoter. And so there's a lot, I think, that we just don't understand. I really think that we don't understand. And the beautiful, as Amy talked about, where it seems to be more linear, I don't know if you would agree, and we can talk about that more. Maybe it's just different in a metazoan. But it's not. Frank Holsteeg, I should have said that, has seen the same. Vichy Iyer has a paper, sees the same. And then people in small studies with a transcription factor here and there, not systematic, also see that, and people just don't talk about it. Now it's on tape.
So I have a sort of philosophical question. Let's say that resources aren't limiting. How should we tackle understanding the regulatory network? Like what set of approaches? How do you tackle that problem?
I think a few things. If resources were not limited, I would want to make transgenic strains for every single C. elegans gene promoter and do RNAi, and then find physical interactions for all promoters as well, by many different assays. So even though, I don't mean to bitch about CHIP too much, because I think it's very important to do all these different methods. If only everybody thought that, but kumbaya. And so I think we need a whole suite of methods that we should apply to many, many different systems and then systematically compare. Now, when I was talking a lot, of course, with Job, as you know about this as well, it's like, what is this indirect? And it's also because we're thinking textbook, right? And so I really think we have to not dismiss indirect by, it's indirect. Well, yeah, so why, right? Clearly it's doing something. And so then we have another dimension in which we have to add all these other different types of regulators. And I think at least what we hope to do is to gain insight into some of those principles, and then people should take their system of interest. It would be very interesting to see if what I was saying about archaea, which may be way not right, if there's differences in evolution for these types of things, which also relates to what Anne talked about earlier. But so measure everything with high quality methods and open your mind, like way open.
So I love this part about the propionate having regulatory effects, and of course, simpler hypothesis would be that it can directly bind to a transcription factor or to an interaction partner of this transcription factor, Were you able to
It's
test
3
this like
carbons.
biochemically?
It's 3 carbons. That's it. 3 carbons. It's nothing. So it's technically very difficult. That's one.
Okay.
Secondly, we thought we had 1 transcription for the longest time. Now it turns out we have 2, and the plot thickens. It could be propionyl-CoA, which is interconverted between propionate, which is unstable. Propionate is volatile. I think so. And Jussi, is he coming? Jussi? I saw him in the schedule. What we're hoping is that maybe we can crystallize it and then see propionate with the transcription factor. But using it, you can't label it. Like I said, 3 carbons. What are you going to do?
So one thing that we've done for plant hormone receptors is just to add the small molecule into the yeast media, and we could see differential interactions.
We've tried that. Didn't work.
Okay.
And we know that, yes. We, of course, think first yeast as well.
Yeah.
One reason is that we know that these nuclear hormone receptors need to physically interact with a mediator that may or may not, that doesn't, I don't think, occur. I am actually not entirely sure, occur in yeast. So maybe you need a larger setup. Other questions, of course, this is what we think about all the time, right? Other questions is, propionate is made in the mitochondria. Nuclear hormone receptors are in the nucleus. Are there other proteins in between? Does it just diffuse into the nucleus?
Diffuse, right? Can go through membranes, propionate.
Out of the mitochondria is probably hard. Into the nucleus should be easy. And then again, how are you going to see if propionate is in the nucleus? You make a nuclear extract, everything leaks out or leaks in. And so these are very difficult questions. And so I think by systematically mapping these networks, we can see if there's anything in between those two. For instance, histones are propionylated. That's a hard word, which nobody talks about really because we don't know anything about it. It's acetylation, methylation, but propionate is the next carbon up, right? And so we really don't know yet. I personally think, which means nothing, that it's a ligand for a nuclear receptor. It's a fatty acid. I really like that ID. I like C. elegans 275, give or take, nuclear hormone receptors. I think these serve to monitor endogenous and like xenobiotic as well as endogenous metabolism physically. Showing it needs to be somebody's project. Would be the cool. I want to show that if it's true. Otherwise, I don't want to show it.
So, if you feed your bacteria differently, how does that change what you're seeing in your...
We haven't done that, except what Auri did in her study is when we've tested 3 chemotherapeutic drugs, camptothecin, 5-FU, and FUDR. And there we see that for FUDR, the animals do better on Comomonas. For camptothecin, they do better on E. coli. And then we've done all this bacterial genetics, and we know a lot of bacterial mechanisms. By the way, 5-FU and FUDR seem to act through ribonucleotide metabolism, not deoxy, if you care about that kind of thing. We can't really explain the dietary difference yet. Moving on to now feeding the dimensions of the problem just explode. But yeah, I would love to do that. And we're going to start metabolomics, of which I am super excited and absolutely terrified.
Let's take one last question back there.
Yeah, great talk. So I really liked the first part where you talked about the coding mutations in the LHX4, and then you also talked about variants that affect binding of transcription factors to their binding sites. And then in the second half of the talk, you kind of shifted away from that and talked about this cascades of transcription factors that regulate other transcription factors. And in the end, you kind of have these leaves that in the end are the physical binding events that we actually should care about.
I couldn't have done a better job myself.
Yeah,
You got it. That's great.
So I'm interested in what your plans are to integrate kind of that information that you have with the cascades of transcription factors and the leaf nodes that are ultimately important for binding and for regulating gene expression with kind of variant information or nucleotide variants to try to understand how different individuals or how the variation...
Yeah, that's a very good question. So we are not yet doing that in C. elegans. There are very few coding or non-coding nucleotide variants give a particular phenotype. What we are doing in a very exciting collaboration, it's also stuff that I barely understand, but in any case, We are starting to do population genetics in C. elegans to look for wild isolates and if they have different responses to propionate. We don't know yet if we're going to find coding or non-coding, but you know, in C. elegans, if you look, all the forward genetic screens that have been done in C. elegans, I'll go out on a limb here, 99% of the mutations that you find are coding, which also says something about regulation, right? There's so much layers and so much redundancies and built-in, and I should say also, I forgot to mention that Martha Bullock also has a very nice study on coding variation in transcription factors and I would call those biochemical or biophysical effects. And then Juan has shown that at the meta level you can see a correlation, but the predictive power of each individual edge, in my opinion, is still lower than I would like it to be. Michael.
One bonus question here.
Hey, how much have you thought about the possibility that that enzymes that actually modify propionate one way or the other are actually, at the same time as they're modifying, signaling enzymes which are affecting transcription factors, which you already have a sensor, then there's a molecule that can bind and then can directly couples concentration to
It could be. I would love to have a way to figure that out. Now, the basis of what we see here is genetics. These propionate pathways, it's like basic metabolism. And so as far as I'm aware, because now I have to compute all this stuff in my head, there's no examples of that in larger metazoan eukaryotes, but why not? Yeah.
And the GAL pathway does it that way effectively. The enzyme can have a signaling function.
Yeah, that would be cool. Maybe you can tell me a little bit more on how we could find that using the awesome power of the worm.
Okay, let's thank Marian again. And let's